So you can see how the edges, sort of that circular edge, is the finer grain material that isn't compacted, so that would easily erode and slip down the slope in front of us. But just digging a little deeper, you see it's a little more compacted, so you can build that tunnel.
I wonder if it's trial and error and they start digging some places and get a whole bunch of sediment on their heads and have to try another spot.
And who knows, maybe these sands have newly deposited on that feature too. If there's an increase in current flow, the grains on the top of the mound here could easily just be pushed over the edge and cover over something that had been previously excavated.
Like another
Canyons basically do feel the effects of bigger storms, nor'easters and hurricanes. And
yeah, that sounds good. Yeah.
Okay, let's do the same.
So we have a question from the audience. Colleen John, how many invasive species have you come across on this expedition, and is there anything being done to limit the results they are causing on the East Coast?
15 meters, 320, 0.
I have not recognized any invasive species.
Yeah, I don't think so.
And probably one of the problems with that is that we still don't even know what is usually found in a lot of these places. You know, we've gone to a bunch of, was that a hagfish? A hagfish right swimming.
Now, yeah. Follow that hagfish.
I never thought I'd hear you say that. Be berries. But yeah, Colleen, in shallower waters, and especially in lakes and rivers, like we have the mussel, which has invaded the Great Lakes region, things like that. Those are easier to track because we had a good idea of what was there, and people are looking at that environment also. But when we come out here, there's such a variety of habitats, and they aren't even well explored yet. So it would be more difficult whether a species or not. But I haven't seen anything that looks unusual.
Do you want me to try and get some light in there?
No, that's all right.
All right.
Thanks.
All right, you're all set. Thanks.
Yeah, the France wrote in that a lot of the marine invasives come into an area because of ballasts from ships and ballast water and or rocks even that are taken up in one place, ballast, and dropped elsewhere. But that would probably be a problem in shallow waters, not in deep waters, because most of those organisms couldn't live in this kind of very different, cold, dark environment. So you have to be very suited for this. Okay. Sure. And in shallow waters, one of the invasive species you hear about, actually not even that shallow, down to mesophotic depths of 100 meters, or not quite 100 meters, but in the upper, barely silent waters, is the lionfish, and that one is quite an invasive, and Casey likes to eat them, and that's a very good thing. You know, there's a a harvest of them. It might help keep the numbers down.
Yeah, and we've really seen their numbers increase along the southeastern seaboard. I was on a submersible dive in 2001, and we saw one of the first lionfish. A few had been sighted. I think our submersible was the first to actually record it on video. And my son and I went back to that same locale and had an ROV several years later, and there were 50 or more in the field of view. It was incredible how much it had changed. Just, I think that was only about 4 years later.
And what kind of depth was that?
That was at the shelf edge, I want to say 100 feet.
Okay.
There was still light, so I know that we were able to see them. just off of South Carolina. So definitely influenced by the Gulf Stream, probably having been transported north by the Gulf Stream from Florida, which is where we think they might have originally been introduced.
Mm-hmm. Pilot, can we look at on the left side? Just went off screen. It looks like there might have been a fish in a burrow.
Yep.
Sure.
That's us.
Let me get positioned over there.
Great, thanks. It looked like a couple of those eel pouts were kind of fighting.
Oh, really?
Just a bit ago.
A pout bout.
Among friends.
Still below off screen, watch lead?
A little bit forward, I think.
Okay.
Just further to the left. Off screen left.
Yeah, and another aspect.
There he is.
Yeah, Siri, there he
Oh,
is. I wonder if this is the same one we saw yesterday that everybody was debating about.
Good eye. Certainly didn't see that.
Oh, darn it. Come back out.
We'll have to zoom in on it digitally.
Yeah. And Scott Francis pointing out that if the larval stages of deep sea animals wind up coming to shallower waters, they could get into the Gulf Stream or a different type of current and travel a long distance. So it might be, theoretically, it might be possible to pick up some deep sea larvae in ship ballast and transport it. And we just don't know a lot about most of these organisms, about their larvae, especially the corals. And that was a barracuda, the little silverfish, which is a midwater
we
fish.
inadvertently collected one yesterday. Did you see that? Did you?
No. Was it on the ROV?
It was on the ROV, yeah. So one of the ROV guys brought it in to see if we wanted to photograph it. And we did.
Great.
Oh, we'd never have been able to capture, of course. So they are very pretty.
You know?
It's only about 7 or 8 inches long.
What? Didn't hear you come up.
I said, see how there's all that sparkle in the water right now?
Yeah.
I wonder if that scales off that silvery fish that just freaked out.
Thanks, pilot. I doubt he's coming back out.
You copy that, watch
out
out.
up there.
Ooh, and here's another maybe octocoral, this white guy.
I wonder if we have not seen, there hasn't been attached to it. We've seen a little bit of that overhang here that we saw in a much thicker sequence at the very base of our, or the beginning of our climb up this ridge.
Go ahead, watch one. Absolutely. Do you want to zoom on this coral, or should I just start traversing? Okay, go on in video.
So I think this is the one that we saw lower, but I don't know if we got a great look at it, so this is really, oh, look at the little, are those eggs? The little pink round things inside.
Oh, yeah.
Yeah, those are at the very least gamete packets of some kind. We were looking at this earlier today. Possibly eggs, but also possibly sperm bundles.
Wow, that's...
Bottom line is that the gametes for these octocorals are produced and stored on the mesenteries, the sheets that divide the inside of the polyp gastric cavity, kind of like sheets of curtains. So this is definitely a colony that is in a well-reproductive state.
Yeah. Did we ever figure out what genus this might be?
No, I wasn't certain. I didn't go out on that limb. I think it's in family Nepheidae, maybe a Scleronephia. I don't know if it's a Drypha or a Duva or something like that. But one thing I will say that's kind of interesting and is useful for us looking here is you can see that not all the polyps have the same number of let's call them eggs for the sake of argument. They don't all have the same number of eggs.
Right.
So they appear to be at different stages of reproduction.
And so that might be a strategy so that they can extend the length of time that they're reproductive and hopefully find a great day with good currents to take larvae away. Or
another possibility is that, maybe what we're seeing are eggs and different polyps produce the sperm, and they're either not as visible or in the polyp. One of the challenges, of course, is that sperm and egg hang later, and many of these species are broadcast spawners, so they'll just release these gametes out into the water column, and ideally you would likely want your sperm to bind with the eggs of a different colony. So it may be that tying of developments as well.
So that they're always ready.
Yeah, that is one strategy in the deep sea, to always be ready. One of the things that I constantly am mystified by when I see these corals that are, you see one here and then we don't see another one for another 10 meters or farther, is how on earth they're able to reproduce with each other.
Yeah, it seems like such crazy luck to have the sperm and egg find each other in the vast 3D ocean there.
Right, exactly. And it's an indication of why they have to produce so much sperm and eggs, so they get that lucky one to combine. And then they have to have the luck that the larva finds a place to settle and have enough food to become a new colony and start the cycle over again. And it's entirely possible, I don't know, but it's entirely possible that this can self-fertilize. So sperm from one polyp could fertilize an egg from another polyp, but that wouldn't result in much genetic diversity far down the line.
Right. So I don't know if we want to eventually take a sample of this species so that we can try to identify it better. But maybe we should leave this one.
Yeah, I'd be happy with that if, I guess this is going to be on a crumbly substrate again, so we might be able to get it that way. Cheryl, is your impression that this is different from the duva that we collected last week? Or
yes, it looks quite different to me.
That's what I think
That's
too.
just superficially, though. I haven't really been able to look at any of it carefully, but yeah, I would say that one was much more brownish, kind of, and the polyps didn't look as big and extended. They looked a lot smaller.
Right, I agree. These are very, very tall polyps. And it might just be the angle, but another thing that was different is that other ones seem to have more branches from the main stalk.
Mm-hmm. Yeah. Yeah, I agree.
So, yeah, I would certainly support a collection of this.
Should we wait to find one that isn't so full of eggs?
The last one was also full of eggs, so this may, although we were talking about continuous reproduction, it may also be a seasonal thing. So it may be that, the summer, there's lots of food in the water column, lots of phytoplankton. We've been seeing how much marine snow is. They might just all be reproductive at this time of year.
Okay, well, maybe since we're set up, we can ask the pilots whether this would be an okay place to try to take a sample.
Yeah, we can go ahead and do that. Watch lead.
Great, thanks.
Okay.
Yeah, and
Full live
Andrea Quattrini agreed that this looks different than the duva that we were seeing earlier on this expedition.
See how close
We
you
saw
are right
that
now?
duva a lot on the coral mounds.
You're going to have a little bit of trouble getting your arm in there.
The Stetson
Okay,
Mesa.
so
You might
back
wanna
up.
Back up like a foot or something. Just
Okay.
enough so that you have space to get your whole jaw and everything down in there.
Okay.
That was a little reddish brown kind of.
Yeah, reddish
Yeah.
brown
What do you think
and
about
Very
that?
fleshy.
more
little bit more?
fan-like. But
Smidge. That's pretty close. Yeah, that's probably good. Something right around there. Nice. And just so you know, when you grab this, it's going to probably do one of two things. One, it's going to try to force all the water out and scrunch itself into a little ball.
Okay.
yeah.
And two, once you do get it, you're going to release all that water, and it's going to, like, deflate.
Okay.
So you just get in there, get yourself all lined up, and then just snag it. Because if it balls up, it's going to be harder.
Okay. And then just kind of close the jaw on it, and then.
I think if you get the jaw and everything right down in the mud with it,
Mm-hmm.
and then close down with the blades towards the dirt,
Yeah. So.
you can still get it in the soft part of the jaw. Just grab it.
So angle the wrist, kind of like level with the dirt, and then come in with the jaws,
Yeah.
kind of at the same slope,
Yeah,
and then.
this is it, like that.
Okay. All right.
Yeah.
Are you going to go from port outer probably? Think about where you want to put it first.
Yeah. Yeah.
You shouldn't have to cradle. You can just
Yeah, that we're sitting still. You
get
can
down
sort of
with
see a little
the
sparkling in the
hose part
water.
of the jaw and squeeze it.
And,
Okay.
I
Jaw
don't know for sure,
lock,
but that
and then,
would suggest to me
Okay.
that
So
those
go
are
hose bottom.
perhaps mineral
yeah.
matter that have come originally from the land.
All right. Do you want to?
It's
Indigenous
pretty small, so I
material.
don't think you're going to get
Those
this
are the
section.
clay-sized
Just grab
particles,
it.
that little micas
Do you mind,
are an
getting mini
example
Zeus on
of
it?
some of the material
Of course not.
that may be washed out to sea, and they have very flat, reflective
Switch it over for you.
faces to them. Also
Want me to zoom in a little
helps
bit
prevent
for
them from settling
please.
easily.
Can you zoom the mini, please?
Or they could be metallic minerals as well. But those tend to be
Perfect.
denser and settle
Lovely.
a little more rapidly.
Good.
The sediment samples I've been collecting, I've washed through a sieve that is 63
Copilot,
microns
would you mind pointing
mesh
over
size,
the arm so I can
and
kind of
that
monitor
allows for
Oh,
the
yeah.
clay-sized
still?
minerals
Light
to be
again on
washed
mini,
out
sorry.
to leave behind the next grain size category, which is called
Okay.
silt, and of course anything larger than that. And it's
Okay,
been a fun
hydraulics going on.
comparison, just preliminary visual comparison. I haven't done any analyses really, but just to quickly look at them under our, it's a microscope with not a tremendously high power to it, so not too much detail, but when we were farther offshore, almost all of that silt size material, just about 100% was
Boy, it's really
planktonic foraminifera with some pteropods and just a few benthic foraminifera.
What's wrong?
Those are
Just,
foraminifera
it's,
single-celled
the viz is
organisms
kinda-
Oh, yeah.
that live. The planktonic ones live in the sea surface, and when they die, their shells settle to the sea floor
Just
and
gonna
make up the
leave
bulk
that
of
that
the
way.
deep sea marine record until you get down to greater depths, because that calcium carbonate material dissolves at greater depths where the seawater is corrosive to
Yeah, so
calcium
You can
carbonate.
actually keep, it might
In
help you
these
keep
shallower
that-
environments,
that-
especially
the
those
wrist
that
up,
are
down.
adjacent
Okay.
to the land,
wrist pitch.
or so close
Okay.
to the
Keep
land,
that down if you
we
want,
typically
I think,
see
or
more of
out a
the
little
mineral
more,
matter that is
and then use just
coming
the-
down
the
through
jaw rotate
rivers,
to get the right angle.
ultimately
Okay. I was just kind of
sourced
thinking
at
it's kind of
maybe
tough
the
to
Appalachians in this area. Some
get
are
in there.
just
Yeah, okay.
sediments that are in
So,
the landscape.
So
but
left, right, looks
basically
good. You're
it's
gonna have to come all the way
terrigenous
left to grab it.
material.
yep.
And those are
Wrist
not the shells
down.
of planktonic marine organisms, so we see a lot more
Okay.
mineral
So now
matter
you're already fully
in
up
that
on
size
the wrist.
fraction.
Yep.
And in the smaller
Just
fraction, of
winding
course,
your
you see
wrist
those
like that
clays I was
to-
mentioning
on
earlier.
the, at the get-go,
Yep.
he's going to end up.
Those forams were
it's gonna
beautiful. I
hinder
loved
you when
seeing
you
them
try
under
to
the
get it
microscope.
I know,
Okay.
I loved getting
So
your
getting
reaction when
it
I
a little bit
showed them on the
unfolded.
screen.
You're going to have to come pretty far left.
Very
but
pretty.
allowing
Little
the wrist
pearls.
to go down a little bit.
Little bundles of
So
pearls.
this inch here down.
Okay.
That should allow you to have a little bit of play in the wrist when you get there to line up. So, I think you
So
were
as a geologist, we like to understand the different environments and see how the sediment record at the top of
So
the,
those are
of
the
the sediments,
grabbers on the
which is the
bottom?
modern sediments
so
usually,
that's the
how
knife
that
edge.
reflects
Yeah, so you
the
want to point
environment.
that towards the
And
dirt.
certainly this
Okay.
environment
Yep.
here is
And there's the
very
squishy
fine
part.
grained, so it tells us there isn't
So-
a lot of velocity
Turn it
reaching
there,
this
that, sort of that
seafloor
direction, so you wanna
environment.
face 'em.
If it were high velocity,
the
the fine
squishy
particles
grabbers
wouldn't
are
have a
towards
chance
us.
to settle out.
And the knife edge
But
is
here,
towards the
it's
sand.
very low energy, so the fine particles settle out. There hasn't been enough transport velocity to bring
Okay,
larger
so
grains
that's
here.
pretty much
Those have probably
the
been
wrist
swept
all the
down
way
through
back
the
right
canyons
now
to
already.
the deep sea.
Yeah, so I was thinking unfold that.
And as we go
You don't want
offshore,
the
you
wrist
see
all
the
the way back, you
composition
want it
change,
down,
and
sort of
as I
straight.
said, the
And
shell
then the
material
left
starts to
should be
increase.
over.
So they tell us a lot about
Okay.
Am I
the
ca- am I
environment
confusing you more than
of their
helping you? I
deposition.
can stop commenting if it is.
No,
more
no, I
confusing than helping.
so
appreciate
that when
the
we
help.
core down into the past and look at sediments that represent thousands to tens of thousands to hundreds of thousands, and then of course millions of years ago, we can start
Okay,
evaluating
so
what a certain location
come
went
in
through
with
in
the
terms
wrists
of changing
like this.
habitat, maybe changing
a little
water
bit,
depths,
and then more
the sea level
down.
has risen or fallen,
So I would
different
yeah,
water
I would bring it in-
chemistries,
I would bring it left
different
a little
biota
Okay.
bit,
have
in more a little
inhabited
bit, and then
a certain
see,
place
so
through
that's the
time.
shoulder,
Yep.
and the
So
elbow,
it really
then the very
tells
next
us
joint's the
change
wrist
through
up down.
time
Yep.
at
See
a
how
location.
that's locked all the way up?
Yep.
That's the one I'm saying unfold.
Okay.
So if you know
The
that for
left
one
coming over,
location
we
and
had
you've been
right.
able to age
We
date
had it
all those
correct
different
maybe.
sediments, you start getting a sense
See, I'm
of
just trying to
climate
get a
change or Earth history
kind of the
change.
jaw
Go back
level
far
with the.
enough,
Right.
plate
So now
tectonics
you,
and the
you
origin
unload
of the
that down a
Atlantic.
little bit,
Yeah.
and then you bring the left
Different
further,
Paths of the Gulf Stream are recorded in the sediment
and
record.
then you rotate your jaws to
So
match.
we unravel a lot of Earth's
Now you
history
have a
by
little bit more
coring deep
play.
into these
on
sediments.
okay.
matching it. See,
And then
now
going
you're-
back in
now
time,
you-
the
as you
closing of
wrist
the
left
Isthmus
and
of
right,
Panama.
you're gonna
That's
match
right,
the
that's
bottom.
right.
Different
Okay.
forams on the
So now
Pacific
you can
and the
Yeah.
fiddle
Atlantic.
with that.
Yep, there
and
used
get
to
it
be
just
an
right.
open
Whereas
connection
if you lock
between
one
the
of your
Atlantic
joints out,
and the
you lose
Pacific,
the ability to adjust.
where
Too
Okay.
close.
Panama is today, where Central America is today. So there was sort of a free exchange of water and a pretty strong current that went through there as well. And when plate tectonics caused Central America to literally rise out of the ocean through a lot of volcanic episodes,
All right.
Tilt you
built
up a little
that.
back up there.
connection between North and
Nose
South
from-
America, it isolated the Atlantic from the Pacific, at least
So
in
what
the
do you think?
tropical
Make a grab for it?
latitudes, and
I don't
began
think that
to
you're close
change
yet. I think you're
who
gonna
lived
need
in
to
what ocean,
wrist
basically,
left
and
keep
so we can
shoving the
see
rest
that
of the line into the
evidence
wall.
in the fossil record
Right now
as
your
we
fingers
drill
are
into
touching.
sediments
Yeah.
on each side.
So your fingers are going to be scratching the wall before you.
Also change
the part that you're trying to
circulation,
grab
which
with is able to grab
if
it.
you study any oceanography,
That
you'll
makes
learn
sense.
how important circulation of the surface waters affects climate and how climate affects circulation of surface waters. So it's a field of oceanography is an incredibly wide area of
You can always close
multiple
your jaws
disciplines
like
all working
partially and
together
see-
to try to
see
understand
where they
our
they are
single
lined up.
ocean
very carefully.
that
There you go.
has persisted through many, many millions of years.
Did you get it?
I'm not sure.
I'll let the dust clear.
Spin your jaw around and see what's on the underside. Is that it?
I think it might be.
Yeah. Okay. You go on port with this?
You're gonna be able to open.
Yeah, I got it.
Cool. Nice grab.
Yeah, I think I see it sticking up towards the center of the manipulator claw.
You going port box with this?
Yeah. So can we get him out?
I think the drawer's gonna hit
And
the
while
wall.
we're
So should
waiting
I?
for the
kinda
sample to be
just
collected
your
here,
index out,
a
right?
little bit
Yep.
more about invasive
So you can just
species.
take the
Les
auto
Watling from the
joy
University
lock-
of
or just
Hawaii
the joy
said
lock
that
out,
Uh-huh.
leave
one
auto heading
place
in,
that we can
just
compare
push backwards on the stick a little, push yourself off the
records from the
Okay.
past versus
And you can
now
set it
are
out of
at
depth if you
museums
want,
such
so
as
you don't
Yale and
sink.
Harvard and the Smithsonian,
The current,
Um,
I think he'll push me in
and
into
that,
the
but
wall.
still
Yeah,
that's
so that's why you give
more
yourself a little
local
bit of a
or
shove
recent
backwards.
times that we can
Okay.
compare. But I
So you don't have to
still
rise
don't know
or lower at all.
if you could just sit
yourself
on the
off.
seafloor
Set
and tell
auto
immediately
depth.
whether something was invasive or not.
There you go. That's plenty of room.
Okay.
When you're ready, I can extend the drawer.
Okay, I'm ready.
Extending. You all right?
Yeah, went into master mode.
Still there.
I'll bring this around for you.
Oh, I think I lost it.
Really?
Oh no, that's a fish.
I think it's still there. It's on the
Yeah.
underside there.
Yeah, you got it.
Oh yeah, the barracuda fish is having a bad time.
That's what I was talking about.
Oh, the siphonophore?
Yeah.
I hope they're going to see lots of those on the midwater transects.
I know.
So I'm going for the inner or the outer? Looks like they're both kind of...
The inner is where I have most of the stuff. Some of it drifted into the outer, so you need to go for the outer.
Should I try and do a starboard?
That would certainly be more, less contamination.
Okay, let's do that.
Okay, let me retract the drawer and open that box for you. Standing.
Then when you could get the mini Zeus over.
I don't think so. Yeah.
Just going to give a bump forward so we don't get too far away.
Okay.
Okay, is that full out?
Fully extended.
Oh, it did?
Okay. Starboard. Inner. You go in?
Yep.
Okay.
I let it go already.
Okay. Great.
I
Ready?
can get a look with the camera.
Do you want to have a peek? You can zoom the mini.
I think D2 needs to wash his fingers.
Yeah? You got any more zoom, or is that all we got? Okay. Okay, let me bring it right over.
Here, pilot. You can still see bottom, so you're good.
Okay.
Oh, yeah, I think it's right there. I'm going to close the drawer.
Copy.
We're letting the water clear in the bio box to see if the specimen actually made it in. It's pretty hard to tell.
Can I get a better look to first
So
tow
we
then?
think we
Go
have
pilot?
specimen
Yeah,
number
it's
2,
coming out
bio.
wide. Yeah, so at this point, if your shoulder's all the way up and in the right
It's
position,
a nice view of the
wrist
steep
and
wall
everything will
here.
just sort itself. As long as the jaws are turned the right way and the
Okay.
shoulder's all the way up.
And
when
I've
you
asked
turn
the
it off,
pilots
it'll
to
relax.
kind of run
Okay. So
along
just
Don't
this
go crazy. Push
little
all those
ledge at the
things.
top
Oh,
here,
yeah, that's a
kind of
good
from
idea.
the side, so
Okay.
that we might be able to
Feel it.
see a bit
It'll-
more.
It'll find its way
Hydraulics
home.
off.
Yeah.
And we are at a depth of 540 meters.
Bring it back over.
We have traveled a whopping 38 meters of relief since we started, but
There's been a lot to see.
yeah.
And we just collected one of the only corals that we've seen so far. We've seen several of them, but this will be a good characterization of this habitat since it's the only one we've seen. And then
Okay, we're going to do
the
a...
scientists couldn't agree on
pilot
what it
change,
was, so
actually.
a very good collection for both taxonomy and site characterization.
Here's another Elix squid. And our colleague Stephanie Bush did a wonderful short video clip, or the video team grabbed the video from her narration of this squid and its chromatophores, and that should be coming out on our website at oceanexplorer.noaa.gov. And just check out the Windows to the Deep 2018 expedition, and you can see our image and video gallery, and I think you'll be quite amazed at the variety of things we've seen and how much fun the video clips are. They're only a few minutes long. They're highlights of the day. Very informative. Some are quite fun and set to music and masterfully produced by our videographer team here.
And we have another pilot switch going on, so
So this little landscape here is a little more hard-packed. Those sediments have been squished together enough so that they are a little stiffer and resist erosion a little more. So you can see little
Good, how are you?
very small little cliffs that some of these organisms really take advantage of.
Understood.
Don't blame me.
And this is what
Go ahead, watch one.
underlies all that drape of the sediment cover that we've been seeing as we climbed this very narrow ridge. Very steep slopes on each side.
Yes, absolutely.
Watch, this is Navigator. So you said that you want to stay on this depth and kind of go around this ridge this way? Oh, okay. All right. Copy that.
or maybe this is something that will run out and then we won't have anything to look at, but we'll try to follow it for a little while.
Copy. There's a fish underneath that soft coral.
Mm-hmm.
Yeah, video we can frame up the coral and fish here.
Nice little caves.
Mm-hmm.
So this row of caves tells me that the sediment that is on their front lawn, that's been there for a while. And they haven't had too much new sediment, or it would have piled up in front of these caves. Of course, maybe they continue to dig out.
All right, Fernando, I got that ridge at maybe a 315.
Since they're all the same
It's
layer,
exactly
I think that
perpendicular
they've
to me
probably
right
been
now.
around for a little bit.
See that?
Yep.
I'm trying to figure out which direction the ship's going to move. Where was the last move?
Oh, and there's a whole
That
bunch of
was,
pycnogonids on the left that
320.
we were just seeing.
Okay.
They like this environment.
They sure
They
do.
blend right in. It's really
I
hard.
see a couple
there's-
here now.
Yeah,
Do you want another move
several.
that way?
The more you look, you more the
Not
fun.
quite yet.
You
Yeah.
find them.
Copy that.
Pilot, can you just zoom in straight center? There's a pycnogonid, just so people who are new to our dive today know what we're talking about.
I wonder if that's something different that we haven't seen yet.
Watch lead is calling you, pilot.
Go ahead, watch lead. Yeah. We get closer video. All right, will you come in?
Nudibranch?
Yes, definitely.
Does
Looks
look
like a
like it's
nudibranch.
moving,
Nice.
yeah.
Wow. Beautiful.
If you have
We
more
are
Zoom,
getting
you
our
can
full of
take
the
it.
mollusks today.
We sure are.
Understood.
So this nudibranch, very close relative to the snails. It's another kind of gastropod. Doesn't have a shell. And you can see all those things that look like tentacles that are arising from the back side of it. Those are called serrata. And you see they have pinkish material inside of them. And those are typically, at least in the shallow water ones that I'm familiar with, extensions from the gut. And in many of these species, they're specialist predators of cnidarians. But they have a real special ability, and that is that they can feed on cnidarians without having the nematocysts fire. Those are the stinging cells of cnidarians. And they're able to channel them into those extensions of the gut right up to the tip. And so sometimes you see little clusters of white at the tips of those serrata, and they would store these unfired nematocysts there, so they use them for their own defense against the next thing that wants to come along and eat them.
So they're borrowing some weapons of mass destruction?
That's exactly right. They call it kleptonidy.
Wow.
Those much larger structures at the head end are the basically antennae, or sensory tentacles, perhaps I should call them.
Is this an amphipod making waves up on the top of the screen?
It looks like it. Kind of like the one that had a rough day with the Ceryanthid yesterday. And or the crab. I think it was maybe the crab that was what finally got him.
Yep.
He's free.
And can we just pan down, or
Yeah, but you can zoom out a little bit.
we want to look at the pycnogonid just under it? So very, very well camouflaged. They almost look like the tubes that are also draping on this mini cliff.
Yeah, and Amanda Demopoulos from the USGS was pointing out that those brown fuzzy tubes could be the agglutinating forams for Foraminifera that we've seen before. And if Chuck Messing were around, he'd probably be very excited right now.
Well, maybe we'll get a few in our sediment sample, because we jabbed into a slope like this.
There could be some hydroids mixed in there as well.
So right there to your left, pilot.
Yeah, it's hard to get
just super turbid
good
water
view of the pycnogonid.
Yeah, we have
you like lasers
too
on
much sediment.
Yeah, that's okay, pilot. I just wanted to point them out for the viewers who hadn't seen them before.
All right,
We've seen many today.
Yeah, today we have.
It's like whenever we have
See if
this
we can
kind
get
of
out of this
substrate, we've been
sandstorm here.
seeing a fair number.
Oh my goodness. We made a mess.
It's very interesting substrate, though.
I don't think that was from you.
Yeah, no. I think that was being very gentle. Very turbid water here.
Gives me a new appreciation for the term hard substrate, because it's all relative.
Yeah.
Here, that's the hardest substrate around. And so it provides habitat for a different group of organisms than those that don't require a hard substrate, which would be the soft sediments that are draping everything else.
I'd be there at 40 degrees. We may want to put in like a 10-meter move.
Yeah, I'm fine with that. 10 meters, 320, something like that.
Yeah, I'm facing like 319, so 320 would be fine.
Okay.
Yeah.
And Les Watling pointed out that the pycnogonids here that we're seeing must be somewhat hairy because they have some sediment on them.
I wonder if that is an adaptation so that they can get sediment to better camouflage themselves.
And going back to the little tubes that we're seeing, Amanda Demopoulos also said that they could be forams in tubes instead of the agglutinating.
Forams in tubes. Wow. I don't study the benthics, so that's all news to me. We did see agglutinated forams, though, attached to the coral rubble.
Right.
When we were down at the mounds on Stetson Mesa. So we did preserve some of those. They look like little tree stalks.
Mm-hmm. Some nice burrows here.
Cool, Puck. Rig my Titan and look it down my right toe, kind of straight down.
Your right toe.
Yeah. Yeah, right there is good. Okay, video coming in partial.
Another wolf eel pout. And I'm not sure if that's some polychaete tube worms below him, maybe. And some more of the brittle stars that we haven't seen the body of a brittle star yet. They just keep waving to us from underneath the sediment.
We did see one fall out of the sediment collection.
Oh, that's right.
So we know it's a brittle star, but I don't know if we actually collected one. We weren't trying to collect one, but they were just within the sediment.
Yeah,
that
but you come wide.
Came
I
up.
try to.
We did collect.
Trying to find little pockets that have less turbid water. Is it? Because I have some wide-angle cameras that I can kind of see where the clearer sections are.
Yeah, and if we want to continue to go kind of left and...
Yeah. We have a move that way.
So all we need to see to round out our mollusks on this dive are bivalves and tusk shells,
like
scaphopods.
Right we're just riding the edge here. This is like the only clear area right now. If I look to port, it's just a big cloud. That's the way we're going. This way.
Les Watling also posed a question with an answer. What's the difference between the continental slope and seamounts? Mud.
He's spoiled with his seamount look, I guess.
I know. And I'm glad you answered your own question, because I would have answered with a much more lengthy answer. So, I'm glad you got right to the point you were seeking.
And hold there. I thought I saw something. Maybe not.
But you are certainly correct. That is one major difference in substrate.
You come back out.
Yeah, I imagine in the Pacific, if you saw a slope like this, it would be covered with corals and sponges and lots of other organisms.
So you didn't actually put a toe down there, did you?
Yeah, very carefully.
Okay.
See that little landslide going off from it? What I've been doing is just like taking my left toe, just barely touching it down.
Are you doing that by driving into the slope, or are you using the down,
into? I got the Z-bias pretty much dialed in. So I think it's just a smidge negative
what is this
right now.
Peeking out? Is that
so I just come in for a landing here. Arrest my lateral movement. Push forward slow, and then I kind of feel
it hit. Wonder if that's our mystery fish from yesterday.
In. So I'm just pivoting off left toe.
Hopefully
you can zoom in,
won't
video.
Disturb him.
He looks pretty grumpy.
It does look like
Copy bridge,
the same thing,
complete.
or something like the same thing.
Grumpy fish.
Well, I'm glad we at least found somebody in one of these burrows.
It looks like he has little nostrils.
Yeah. I don't think it
Aren't
is the
those an
same as yesterday.
eel-like fish that have the nostrils?
Yeah, the nostrils, because there's not that little angler fish sensory organ. Yeah, I think you're right. It'd be interesting if all these caves were from this one type of organism.
Right.
Hey, video, you can come out when you're clear. I saw a white subject up above here. Might be of interest. Where was it? There it is.
Oh yeah, there's another white coral and a really nice pycnogonid. Oh, and he's on the move.
Frame up the sea spider.
He's quite a bit bigger than the other ones.
It seems like it, yeah.
Yeah, you can see another one right next to the white coral for comparison.
There's some smaller ones on the left.
If you want to get lasers, you could.
Coming on lasers. Lasers on.
Yeah, he's about 10 centimeters across.
It's the daddy long legs of the pycnogonids.
All right, let's go for sea spider.
Coming off with lasers.
Sounds good.
When we pull back out, you'll see on the right side there's a white octocoral, and it might be what we collected, and it's a better view that'll show that, in fact, it is not a soft coral.
I have video. You can approach.
Scott, does the
Does
the sea spider have eggs, or is that what the bigger things are?
Yeah.
Yeah, I can't tell. The color is different. Maybe it just means they're more developed.
Right.
I couldn't tell if those were egg masses that it was holding or structures on the rock below it.
Yeah.
Yeah, so you recall maybe 2, maybe even 3 dives ago, we saw another colony just like this tucked into a similar position, and we were questioning ourselves about whether it was a white anthomastus-like animal.
Right.
And decided later that, no, this is probably something like Eleutherobium. And I think that may be what we collected, which is great.
Okay, that's wonderful.
Yeah, so fantastic. And Andrea had earlier put in the chat room that Kathy McFadden, who's at Harvey Mudd College in California, and who's an expert on the soft corals, including the nephtheids and alcyoniids, said that last one we collected did not look to her like a nephtheid. It looked like Eleutherobium. So that's fantastic. She was able to recognize it from the view we were seeing.
Oh, that's great.
Yeah.
And this one almost had an amphipod for lunch. Bounced off one of the tentacles and it tried to contract and catch it, but
it just jumped off in the distance too. Can we get any tighter zoom on the coral?
Video, do you have any zoom left? Max zoom, copy.
This one appears to have far fewer gamete development than what we saw earlier.
Right. You can see a little bit of pink, but definitely not like the other one. And going back to that fish that we were just looking at, Andrea does think that's the same one that we saw yesterday, and there's still a debate going on about the identification. It could be a toadfish or a wry mouth is what they're thinking. But it would be a depth record for either one of those species.
Oh, really?
That's a big cave, huh?
Yeah, that's an interesting looking cave.
Want to try the fog lights?
Coming on with fog lights video.
Might not help.
Yeah,
with so much particulate in the water column, the brighter lights don't really help. Kind of like driving with your high beams on in the fog.
Yeah, or the snow.
Yep.
All right, I'll keep moving down the line here. You could come out if you aren't already. You might be zoomed all the way out. Okay. Could probably get another 10-meter move in.
Yeah, I was going to say, as he pushes out.
So yeah, we are just off of this ridge on the south slope, or southern facing slope.
Bridge
Oh wow, there's a Humidita picta. We haven't seen any of those in days now. The squat lobster that's in the little V here.
How is that different from the squat lobster we saw earlier today?
So these are chirostyllids, so they are in a very different group than the what we were seeing is probably Munida, and so they're in the Galatheidae, and this is in the Chirostyllidae, so two different branches of squat lobsters.
In terms of their morphology, what's the distinction? What's the difference in appearance?
I'm not sure we can see that kind of detail, and I'm actually not even sure what it is. I just know what the species look like.
Oh, okay.
So
it looks just like the one we saw earlier to me, so that's why I was curious how I could better identify them. Because we saw a lot of these earlier in the expedition.
Yeah, it was funny. We didn't see them on the Stetson Mesa, I don't think, but it was the Cape Fear site. We saw lots of them.
All right, video, pilot's clear.
And the black-belly rosefish, too. We haven't seen that today until now.
Oh, I guess we haven't. I'm so used to seeing them
right.
on other dives. You're right. We haven't. Very, very common in other environments we've visited, including rocky slab areas that were shallower. We saw them in coral mound areas, and then the last couple days we've seen them just on broad sediment slopes.
And those were quite small that we were seeing yesterday.
Yesterday's,
thinking that could be maybe some juveniles or...
What do you got bailout at, Fernando?
Southeast.
Understood.
Well...
So if that was to happen...
Yeah. Approaching now. What was that, co-pilot?
If that was to happen and we'd want to get into tow configuration, I'd swing to starboard and you would swing to port.
And we have a sighting of a burl starfish that's not buried. The rest of them that we've seen so far have been buried.
You can frame it up video.
And Tammy Frank told me this, and I knew this too, and I forgot. They're actually, the Eummunida squat lobster that we were just looking at is now in its own family, the Eummunidae, while Gastroptychus and Europtychus are still in the Chirostyllidae. So that's something about taxonomy. It is constantly evolving as we learn more and figure out whether molecular work agrees with traditional taxonomy, and or can help guide a more natural classification system?
Cheryl, I don't know if it was remarked on, but when we pull out from this brittle star, about 2 o'clock from this, there's something, and I couldn't tell if it was a sea cucumber or a tube of a cereantid or some other mud.
Okay, we'll take a look.
Alright, video. I think the pilot's clear. The video's clear.
Yeah, there's something at 2 o'clock right next to the fish there. Big long tube looking thing. On the right.
Could come in video.
It may have been, like a tube for a cereantid anemone that's become unburied with the slope giving way.
Yeah. Interesting whether it's still there or not.
Yeah.
And then there are...
Yeah, it's only a tube, yeah. And you can see one next to it that has the tentacles coming out.
Yeah.
That one to the right, or several of them to the right, those might be polychaete tubes.
That's what I was thinking also, yeah.
Maybe the bigger one is as well. Maybe they're all polychaete tubes.
And then we have a few brittle star arms on the right there. And Amanda Demopoulos said that, I guess Scott and Amanda raced to this identification of terebellid-like polychaetes because of the spaghetti-like tentacles that are coming out.
Hour and 20 minutes.
Yeah, these polychaetes are basically feeding the same way as the spoonworm that we've seen now a couple of times that sends its proboscis out along the surface of the mud, but in this case they have a whole lot of tentacles tangled up, but they're basically doing the same sort of thing. They're sticky, they can be elongated, and they can carry organic particles back towards the mouth.
That looks like a very good way to feed in this environment.
I'll just let you move a little bit more, and I'll clean another move.
Understood. Sounds good to me.
A lot of what I believe are jellyfish up here in Sirius's view.
They look pretty, most of them look translucent, or they look pretty red?
Pretty red.
Yeah. Pretty solid. I think I saw one on D2's camera earlier today. I don't typically see those.
So, pilot, are you slowly rising, or?
Roughly the same. I did descend like a little bit.
Okay.
Since we started this transect, but roughly staying the same
depth?
Yes.
Okay.
Just trying to follow this ridge feature here. Yeah, he hasn't moved the winch very much.
No, not at all.
And we're at 18, so I think we're pretty much the same.
I'll be there at 5.5. Probably when I get to 4.0, we can have another 10-meter move.
Okay.
Yeah.
Copy that.
Would it be possible to get a quick zoom here on the little pink things?
Yeah, possible bubblegum come in video.
It is a baby Paragorgia, isn't it?
Wow, that's quite a find.
That's very cool. And pilot with the ID before I even ID'd it. That's awesome. That is great.
You know, it's so small that I don't know that it's a Paragorgia, but full credit to the pilots, it could be.
Yeah, very well could be.
I think there's another one to the left.
Doesn't it?
Yeah.
Wondering if it's these